Chapter 23 is where we're going to start with genetics. So we're going to look at the patterns of gene inheritance in this lecture, and we're going to cover, first of all, Gregor Mendel, who's known as the father of modern genetics. He was an Austrian monk, and he did experiments with pea plants. So he investigated the inheritance at the organisms level. He did this in the 1860s. So this is a process that happened quite a while back. He concluded that plants transmit distinct factors to offspring. We now call these genes, those factors are called genes, and they're found on what we know as chromosomes. In humans, chromosomes come in pairs, and they're called homologous, and we learned this when we talked about mitosis and meiosis. One member of the pair is inherited from the mom, and the other is from the dad. If you'll remember from the chapter before, that homologous pairs of chromosomes, both of them have the same length and the same centromere location, so they're easy to identify. They both carry the same genes for the same traits in the same order, but the forms of the genes along the homologous chromosomes are called alleles, and we're going to talk about alleles such as whether the plant is tall or short, whether it has flowers that are purple or white. So the gene is for the color of the flower. The allele would be its form. Is it purple or is it white? The gene would be for the height of a plant. The alleles would be either tall or short. So we have a look at a locus. A locus is the area or the loci of a gene on a particular chromosome. And if we look at this, we'll notice that this chromosome, the red one and the blue one, are identical. They have the centromere locations that are the same. The only difference is their genes, while being at the same place on the chromosomes, they have different alleles. So this gene here could be for color of the plant. This chromosome carries a capital G, which we'll talk about dominance and recessiveness here in just a little bit. The little g would be the recessive form of that allele, and you'll notice that all the way down through here. Mendel developed a couple of laws that are important in genetics. The first is the law of segregation. This law says that each individual has two factors for each trait. The factors separate or segregate during the formation of gametes. So this is in meiosis that this occurs. So they separate during meiosis, and each gamete has one factor from each pair of factors. Fertilization is going to give a new individual two factors for each trait. So while these factors are separated during meiosis, forming gametes, when fertilization occurs with a sperm and an egg, then that number is going to be doubled again, or come back to the complete number. For humans, it would be, instead of 23 individuals, it would be 23 pair, and that allows that individual to have two factors for each trait, one from mom and one from dad. The inheritance of a single trait. We're going to talk about phenotypes. Phenotype is a physical appearance. So an individual's actual appearance or physical appearance, and that's the way I try to stress to help you remember that pheno is physical, is the appearance visually inspected. A genotype is genetic makeup. So the alleles the chromosomes carry that are responsible for a given trait make up the genotype of an individual. So you can't look and say, oh, that person is big A, little a, or big T, little t, or big T, big T. You can't see that. You can see the phenotypic response to the genotype being what it is, but you don't always know the genotype of an individual. There are two alleles for a trait, one on each chromosome. We are going to let a capital letter symbolize a dominant allele, so this is a capital W, and a lowercase letter is going to symbolize a recessive allele. And I want to show you something here that we're going to use to help with our note taking. You notice that a capital W and a lowercase w are both the same, only size is the difference, and we all write in a hurry sometimes, or write differently, and it's hard to keep up. If I'm going to do a capital A, I'll write a capital A, and a lowercase a would be written like this. Those are an obvious difference. When you're talking about S's, or you're talking about W's, or you're talking about C's, those you need to make sure that you identify the difference between the dominant being capital and the recessive being lowercase. So what we're going to do to note take is to put a line over the W. Let's just go over a couple more examples real quick. Let's say that the individual I'm talking about is two lowercase c's, so recessive c's. So the way I would write that, if I write it like this, you don't really know unless you're comparing with the other letters around it, whether they're capital or lowercase. So if I do this, then you automatically know it's two lowercase c's. Same thing with s's. You can write it like this. So this is a capital S and this is a lowercase s. So just remember that note taking your lowercase, if they look identical to the uppercase but just smaller, we're going to put a line over the top of them so it'll be easier for us to keep track of. The dominant refers to the allele that will mask the expression of the alternate or recessive gene, or allele, I'm sorry. Now, a dominant personality should be something that you're pretty familiar with because you have those friends that it doesn't make any difference if you're in the room or not. They tend to kind of take over the whole room. They dominate the room. So they cover up whoever's with them by not allowing them to be seen or paid attention to, I guess you could say. So a dominant allele is one that you only have to have that one copy of a dominant allele in order to not see the recessive at all. So dominant takes one copy in order to express it. With recessive, it's going to take two to express it. Here's an example. So you have one to go off of. We're looking at the widow's peaks here. So you see this really defined widow's peak, and we tend to see these really evidently in males because they tend to have that receding hairline that goes on a little bit quicker than females. So this evidence of a widow's peak is this V here in front, and this is a straight hairline across the top, no V here in the middle. So if an individual has a widow's peak, they are either big W, which is homozygous dominant. Homo means same, so big W are the same, and it's dominant, they're capital. Or they can be heterozygous. Hetero means different. So big W, little w, the big W, one copy of that, making the widow's peak the phenotype that it is displayed. So when we look at this gentleman here with the widow's peak, do we know what his phenotype is? Well, his phenotype is evident. He has a widow's peak. But we don't know his genotype because he could either be big W, or big W, little w. We can't tell by looking because it only takes one copy of that gene to make that evident. On the other hand, the woman that we have over here that has no widow's peak, a straight hairline, is homozygous. Again, homo is same, so big W, homo is same, little w, is homozygous recessive. We don't show the dominant trait at all, and we have to have two of those recessive genes in order to see the recessive trait in this case. All right, let's talk about gamete formation. During meiosis, homologous chromosomes separate, so there's only one member of each pair in a gamete. So if you remember from going over the process of meiosis, let's say that dad has a chromosome that has a big W on one and a little w on the other. When that goes through meiosis, you're going to end up with one big W in one cell, because it's going to be pulled over here, and the other, which is the little w in this case, in the other cell. So we end up with two cells, two possible sperm cells, one having a big W, one having a little w in it. So there's one allele for each trait, such as hairline and gametes. So if the genotype is big W, little w, that means that one came from mom, one came from dad, and if this individual goes through meiosis and makes a gamete, they are either going to give this big W to their offspring in the sperm cell or the egg cell it's producing, or the little w. So it can pass on either one of those in the process of fertilization. So that's going to lead us into, and I'm not going to go into how to do this yet, but that's going to lead us into the crosses that Mendel came up with and used a Punnett square for, and you've heard about the Punnett square before, because I think you did those in like 7th grade or so, how to determine what the possible offspring could be given the genetic makeup of the parents. That's what we're going to get into on the next lecture.